A polypeptide is an amino acid chain that runs in a single direction. Polypeptide bonds bind the amino acids together covalently, and these polypeptide chains go on to form proteins that are required for tissue and organ growth, structure, function, and regulation. This necessitates an understanding of polypeptide synthesis, which is simply the process of creating a polypeptide. Transcription and translation are the two key steps that occur during this time. The nucleus of the cell, which also houses the DNA and transcription site, is where polypeptide synthesis begins. The backbones of DNA are made up of phosphate bonds and a sugar termed deoxyribose, which is followed by 4 nitrogen bases connected by hydrogen bonds. To begin, the helicase unwinds and unzips a segment of the 2 strands of DNA to start polypeptide synthesis. One of these strands will be employed as a DNA replication template. This tiny region of DNA has a start and stop signal that is utilized to recognize the coding for a specific protein that is required at the time. The bases of the chosen DNA strand are linked with complementary RNA nucleotides. Adenine and thymine nucleotides A and T, as well as cytosine and guanine nucleotides C and G, form a pair. RNA, on the other hand, substitutes uracil U for thymine whenever it adds a complement to A on DNA. The RNA polymerase enzyme then binds these RNA nucleotides together to generate a new single-stranded RNA. Messenger RNA (mRNA) is a new strand that contains specific codes and information about the exact order in which amino acids must be linked to produce a polypeptide. Once developed, mRNA goes from the nucleus of the cell via the nuclear membrane and into the cytoplasm, where it attaches to a ribosome made entirely of rRNA or ribosomal RNA, kicking off the next phase of translation. Many tRNA molecules, also known as transfer RNA, are present in the cytoplasm and carry one amino acid, which is the monomer of a polypeptide. The ribosome reads the mRNA code 3 codons at a time in the cytoplasm and adds the complementary anticodon located on one end of the tRNA. The opposite end of the tRNA contains the related amino acid, which is essential for polypeptide production, and these codons provide information that identifies the linked amino acid. After the first tRNA and amino acid have been connected, the next tRNA carrying another amino acid will move in and bind its anticodon to the monomer's corresponding codon. Before the first tRNA and its anticodon separate, the 2 amino acids carried by the tRNA will form a peptide bond, leaving the amino acid behind to be picked up by another tRNA. The mRNA will move through the ribosome, allowing the next tRNA molecule to arrive. As additional tRNA anticodons match the mRNA codons, the amino acid chain lengthens.
This will continue until a stop codon, a codon that does not code for an amino acid, is encountered. Once the stop codon is read, the amino acid chain detaches and the polypeptide is formed.